Hi, welcome everyone to the virtual brain and clinical research, an introduction. And this is week 8, and we will be talking about in silico optimization of deep brain stimulation. I'm posting again the link in the chat for the eBrains collaboratory drive, where you will find the material of today's lecture. So you have the PDF of the slides available, and you have a notebook for our hands-on session because you will optimize deep brain stimulation in silico yourself today. And yeah, we have roughly 2 hours. I'm Jill. I'm a postdoc in the brain simulation section led by Professor Petra Ritter. I have been there since shortly before COVID, so I can always remember. It's 3.5 years now, a little overlapped. And I've been learning only then 3.5 years ago, the virtual brain. And I dove right in and I said, let's do something cool with it. And let's try to optimize brain stimulation with the virtual brain, which I think is a very promising direction for the virtual brain in clinical research. So it's important we talk about it today. And I hope I can inspire you there somewhere in the world, that you also think this is a promising research direction and that you join us in pursuing this huge goal of bringing the virtual brain into the clinics. And a short note, please, already now go to the eBrains collab of this course and download the material, because eBrains is a great system, but will have maintenance from 5 p.m. onwards for approximately half an hour. So that will mean that maybe during our course time, shortly, the eBrains collab will be unavailable, as will be the eBrains lab. So that should be no problem for us. I have also a local setup. Maybe you guys have also already TVB locally. So just download, be sure that you are safe and can follow along, or just relax, sit back and follow along later if you like. All right, let's get right into it. Brain stimulation. The picture on the left is something you have most probably seen in some kind of horror movies. Old movies, they often show this brain stimulation shock treatment as something brutal. And actually it's called electroconvulsive therapy. Sorry, electroconvulsive therapy, and short ECT. And recently it has had kind of upcoming trend again, and it's saving the lives of depressed patients currently. So it's really something that was displayed in movies and got a bad reputation, but brain stimulation by itself is a promising treatment for many people who are resistant to other forms of treatment. So that was then and now. And here on the right side, probably you all know that as well, is a new development of brain stimulation that looks very different than on the left. This is Neuralink, the company founded by Elon Musk, and it's very controversial, to say the least. And it's developing as well a device for brain stimulation. And by the way, if you have any questions, feel free to use the chat at any time. You can also talk rather in the breaks, of course. Otherwise, you are on YouTube, as will I be later on. Okay, and yeah, what to say about Neuralink? It started with a pig. They implanted this device in a pig's head and showed that the pig can be kind of controlled by this device. It's based on some, a round thing, and then there are actually many fibers going into different directions inside the brain that can control different and stimulate different parts of the surrounding tissue, and creating with that different, for example, movements in patients. So recently, you see, it's only been roughly 1 month since they started their first call for their first in-human clinical trial. And it's open for recruitment. And they're searching, for example, for patients paralyzed, suffering from amyotrophic lateral sclerosis, that are willing to get this implant and to join their clinical trial on it. If you want to know more, there's so much out there about different forms of brain stimulation. I just wanted to give two examples so we can have a kind of an overview on what's going on here from historically shock treatment to now, this is something that at least the founders of Neuralink envision that everybody has this kind of, they call it Fitbit in your skull in the future. And Elon Musk even shared his plans that he wants to combine this device with AI technology. So we are humans are not left behind. So somehow in the future, this will be a thing. And let's see how people are modeling it. And let's see what we can learn about it today to not be left behind. So this overview of the whole lecture. First, I will talk to you about the specific form of brain stimulation called deep brain stimulation. The name gives it away already. It's deep inside the brain. But before I get there, I will make a small detour and tell you also then a little bit about brain networks and the virtual brain, and give a bit of heads up just in case you missed the last lectures or you want a refresher or anything in that direction, because I always think these lectures should be, comprehensible also if you join at a later time point, or if you are not 100%, on what is TVB. So let's revise that shortly in between here to then come, how do we simulate deep brain stimulation? What would we have to do to simulate the influence of such an electrode on the surrounding tissue and brain regions and their connections? So we will start simulating it in a mean field model. And that is what we will do as a hands-on session today as well. So you will be simulating deep brain stimulation yourself. Fingers crossed that will be outside the maintenance window, otherwise locally. And then we will make this brain simulation more and more realistic. And we will start with integrating it with TVB multiscale. So zooming in on the tissue surrounding the electrode, giving their single neurons, single spikes, and watching what happens if we give a stimulus. And then we will talk about how we've actually already improved this kind of this form of simulation, and how we're planning to improve it and make it more and more realistic in the future to bring it towards clinical use. And then last but not least, we'll talk about what other neuromodulation therapies are there out there. There are many, as you've seen, and how can we actually simulate those as well with the virtual brain. There will be a break approximately here. So we will just stop after 1 hour and make a 5-minute break, I'd suggest. All right. And then the question is now first, what is deep brain stimulation? It's the long version for short DBS. And it's really deep, as you can see. It's an electrode that actually 2 electrodes on both sides of the head are implanted during a surgery in a patient's head. And they're emitting some electrical pulses to the surrounding tissue. And by that, they're altering the activity of actually the whole brain, as we will see later on. So it's quite a powerful spot where they are. They are deep inside the subcortical structures of the brain. And this is an electrode here, and which is often a target spot is the STN. That's a short abbreviation for subthalamic nucleus. It's the basal ganglia, which is the most prominent target region for DBS. So we have here the GPI, which is the globus pallidus internus, the GPE, globus pallidus externus, and sometimes even the thalamus is the target for deep brain stimulation. And you can see these electrodes actually look a bit like 2 chopsticks, but they have cables implanted as well. So Neuralink is supposed to be chargeable via just holding something onto your skull and kind of a Bluetooth charging station. But this technology here, deep brain stimulation, still has cables, which are in a second surgery put outside your head here. And then they're going all the way down to the chest. And in the chest, in a surgery 2 weeks later, there will be an implanted kind of with batteries, kind of like a pacemaker implant, which can be then recharged by changing the batteries currently. And this is how it goes. It's also nowadays possible that these electrodes do not only emit electrical pulses, but are also able to sense what is happening in the brain and record brain signals. So for that, also this device becomes very elaborate then, and you're able with your smartphone to look at your brain signals coming from this device. So anyways, that's really sci-fi, right? But what is it used for? It's a treatment option for many, many different diseases. It's an established option because it significantly improves the symptoms of Parkinson's disease patients, dystonia patients, and essential tremor patients. So when you go on YouTube, you look for deep brain stimulation, you can really find these beautiful videos where a patient has this DBS device on or off, and it really changes all the movements that he makes. It's quite powerful, but yeah, we don't understand it yet completely. So that's where modeling can come in and hopefully kind of open up the brain and show us what's behind this powerful mechanism. Further investigations have been for obsessive compulsive disorder, depression, Tourette syndrome, Huntington's disease, addiction, Alzheimer's disease, pain. So you already see that there's a long list of other brain disorders where this has been investigated and where this technique has come and shown some effectiveness for relieving patients of certain symptoms. So we must say it's highly invasive, right? So it is a surgery that patients consent to, right? And that's a big step. It carries also a lot of risk, not to make this more beautiful than it is, right? So here we have kind of the idea behind what we are going to do. This is the virtual brain here in the middle, and there we have the disease brain and the healthy target brain. And we hope to bring in this kind of three-dimensional space of brain dynamics, we hope to bring the disease brain via DBS closer towards the healthy target brain. And we are going to use a virtual brain to kind of virtually heal the dynamics of this disease brain. So how does it look when we implant the electrode? This is an average of, I think, 20 patients. And you can see that the electrode is supposed to go into the STN, subthalamic nucleus, and into the motor part. But often after a surgery, when we reconstruct the location of these electrodes, they're kind of all over the place. And actually what has been found is that when an electrode hits and we activate, is placed such that we activate the red fibers, we see a clinical improvement of Parkinson's disease patients. So they're feeling better and their motor, so feeling better, I have to clarify, because sometimes there are side effects. So they're doing better with their motor symptoms. But when we activate blue fibers, actually it can get worse for them afterwards when the DBS is switched on. So we see that really these millimeters apart placements are important and make a difference for the patients. So we know it's a powerful technique. Its underlying mechanism is not understood. And there are already many models out there modeling deep brain stimulation. We have single neuron models, we have whole brain models, but we set out and said, okay, there is not yet a model that combines these small scale models with a large scale whole brain insight that we have about deep brain stimulation. So we set out to build a multi-scale model to bridge those different scales. And what is our vision here? The whole vision is that once in the future, far, far, far in the future, right, a Parkinson's disease patient with some tremor here will come in, get his structural MRI and his functional MRI, and we can build a virtual brain model of this patient, mimicking his or her activity. And then we can take this model, and inside the model, in Silico, we will use some high performance cluster setup pipeline to test and optimize his brain dynamics, bring him or her closer to the healthy state with, for example, here, the virtual brain. Such that in the future, we have a personalized treatment plan, including what settings should the electrode have, where should it be placed, and also on top of that, what kind of medication should we give next to this electrode therapy. So with that, we come already to the next point of how should we simulate it. And we come to TVB. So let me start all the way in the beginning, because maybe you were always wondering what a brain network was. So here it is for you in a very simple display. Sorry, I heard some noise. Is there a question? Please go ahead. Okay. Write it in the chat, or let me know now or later. All right. So generally, we're talking about networks. Network science is the application of graph theory, which is a field of mathematics, and it's rather old, coming from Euler. And it's all about dots and lines. And those dots in graph theory are called vertices, and those lines are called edges. And once we apply it, and here we definitely apply it to the brain, they are no more called vertices and edges. They're called nodes and links. And that's the terminology of network science, which is the application of graph theory. So here we have that a certain region, a node, for example, the motor cortex, every region gets such a dot or node. And then they are connected and getting a link in between them when we find through MRI a white matter fiber tract that connects those two, or often when we find at least a few of them, so that we can be sure that there is indeed some connectivity structurally between those two regions. So I already said structurally because there's another brain network. We have the structural brain network coming from diffuse and gender imaging, following little water molecules through the brain, which we can see as a road network. But we know that once you know the road network, you don't know yet where the problem is and where will be the traffic jam. You can suspect where there will be the traffic jam, but it's even easier if somebody tells you also where is the traffic and what's the traffic flow network between two cities, for example. So this traffic flow from the brain we can measure with time series data of different modalities, for example, fMRI bold data, MEG data, EEG data, and this is how we would then build our functional network. So I always see it as this multi-layer network of two layers. We have a structural and a functional network. And to know then where TVB will come in, it will be our link. It will be based on the structural network, and its output will give us a functional network. So yeah, just consider road networks, traffic flow networks, or any other network where the structure is not everything, and you need the dynamics to get the whole picture. So again, fiber tracking on top of a DTI gives a structural brain network, and we need a parcellation often in between, which is just the division of the brain into different brain regions. And then we have also, for example, fMRI bold data, which will give us the connection functionally, so kind of the communication between two brain regions in a different kind of network. And now, what is brain network modeling? So we have the same structural network, and now all we do is put dynamic models on top of it. So once you understand the structural network and where it comes from, the so-called connectome, structural connectivity, weight network, structural network, there are different names for it. Once you understand this, all you need to do is place a dynamical model that describes the dynamics inside this region, this node, on top of every node in our network. And then we will scale this connectivity here with our global coupling factor, which is just the one scaling factor. It can be like 3 or 4, and that will multiply and scale up or down all the connections of the whole brain structure. And inside such a region, these dynamical models here, for example, from Deco et al., will contain different populations. They will not be single neuron models, but rather what we call neural mass models. Mass is clear because there are many, many neurons in one region. So we will not describe every single neuron, but we will describe a whole population of neurons. So in this model, we have two populations inside every region, one excitatory, one inhibitory, and their interactions are described as dynamics over time. And how we display dynamics in mathematics, we just love to write equations, right? I'm a mathematician myself. So I can tell you mathematicians love equations, and they even more love differential equations. Why? Because differential equation is nothing more than describing what happens to a certain state variable, for example, the firing rate over time. And when there can be like one beautiful equation, very simple, and then when you start simulating and you start looking at the changes that this variable can take, then it becomes beautiful, and you can see beautiful dynamics. For example, like birds through the sky, or you can see synchronization of elements or desynchronization, pulsing, phase lags, and beautiful phenomena that we see in nature, all based on a single, or in our case, actually often more like 6 differential equations. All right, so you place such differential equations, more like 6 of them, on top of every single node. You connect them with a global coupling, and what you see is brain dynamics. This is how simple it can be when we talk about brain network modeling. And the whole approach behind not looking into every single neuron, but rather looking into a neural mass or a whole population of neurons, is that once you track not every single bird's movement, but rather the average of their location, which is around here, I hope, then you can still see where the bird swarm is going. And you can still trace its more like global movements. So that's the motivation behind it here. And when we look directly now at the virtual brain, we see the structural network here. We see there are actually 2. And yes, for the virtual brain, it's often needed that you have 2, because in our models, we don't only connect them to each other via the global coupling and kind of incoming strength, but we also consider that sometimes this link between the 2 nodes is rather long, like from the basal ganglia up to the motor cortex, a couple of centimeters. So what do you have then? You have a delay. And this kind of delay is expressed in the form of this distance matrix between the 2 regions, or we call it sometimes tract length matrix. So it's really the length of the tract between 2 regions. And this length can be something, we often write it in millimeters, right? Can be like a couple of millimeters. And that will tell you when a certain signal from one region to the other region will arrive. And this is as important for the dynamics, because we have seen the influence of these delays on the dynamics is very important. And different delays, for example, in diseases, can lead to abnormal brain dynamics and to sickness, right? And different symptoms. So what comes out of the virtual brain is really what you choose to come out. The different so-called monitors. The local field potential would be just a raw neural signal of the virtual brain. And then you have MEG or EEG you can choose. And you also have a monitor that gives you out of the virtual brain directly the bold blood oxygen level dependent signal, which you have in functional MRI data. So you can kind of look at these equations and their interactions with the virtual brain. So that's it, that's all. And this is a global differential equation then. And as I said, don't worry, differential equations are just equations telling you about state variables over time. In this case, xi, let's consider it's a firing rate. And then we look at the derivative of it. And we have a neural mass model that will give us, once we give in the state at time point t, it will tell us what's the state at time point t+1 internally. But also we need some external influence from other regions. Those come in here. we have the global coupling scaling factor, often denoted as G, that is scaling up or down all the connections. The connections are in a matrix called SC, and these are all the connections and their strengths between region X and other regions, and or in this case, region I and other regions J. And there's a delay as well, because their signals will only arrive with a certain delay in time, depending on how long the track length is. And then we have, sometimes we can choose if we have it or not, a local connectivity, which will give you the connection between a region and not another region that is connected with white matter fiber tracks, but the connectivity between a region and its direct neighbors in the vicinity of that region. So really meaning, like, if we look here, then right next to it, because we know your neighbors have an influence on you. Maybe you don't have a white matter fiber tract with them, but we know in the brain it's quite influential if two regions right next to each other are doing different things. So this is how you can account for this short range connections just being next to each other. And then we have the injected input. That will be the topic of today, which is injected input in terms of injected current, which is something that can come out of a DBS electrode. And of course, always some noise. So I think we will skip over beautiful examples of the virtual brain a little bit, maybe short, okay, short, because I always find them good for motivation. We have an ongoing clinical trial with planned to be 400 medication refractory epilepsy patients going on in France, a different multi-center trial, where patients that are going into resection surgery because their seizures will just not stop are also looked at with a VEP, a virtual epileptic patient. And then that will be discussed with the doctors, which are the resected regions that should be stopping the seizures for individual patients. And they will look at whether those patients are then, after the surgery, seizure-free or not seizure-free for a year here. And these are the preliminary results of the so-called EPINOV trial, where you can see that with a virtual epileptic patient, it looks different than the currently used method. And how does it look different? It seems that something is going to zero here. And what's that? It's for the group of seizure-free patients. We have the strongest effect here, where we have patients being seizure-free after a year, and the false discovery rate, that there was a mistake done and that the wrong region was taken out, is going towards zero with a virtual epileptic patient. And the other method is the so-called epileptogenicity index, which is currently used. So we see some promise that this trial will be kind of a milestone in the move of the virtual brain towards the clinic. Yeah, there's more to come, I think. And here we have, again, our overview, where I just wanted to widen your perspective on these kind of interventions that we can model. And you have heard already that pharmacological interventions like memantine can be modeled with the virtual brain by Leon Stefanowski in an earlier lecture. And also we have other colleagues working on TMS modeling on the virtual brain as well. So basically inside the computer, we can do whatever we like, right? And that's the great part about it. You can see again the main result, that when we give to the virtual brain some memantine, we see that the slowing of EEG signals reversed in Alzheimer's disease patients. And the tumor resection going on with the virtual brain, I think just last week you had another lecture on how one would go about lesions inside the brain and the virtual brain, and here it's about tumors. So it's difficult because the pictures you get are not so nice. You have to do lots of pre-processing, etc. And then you can see that our colleagues from Belgium here together with our colleagues here, Micha and Petra, have already worked with virtual resection of brain tumors here and analyzed also there with a brain network model the outcomes for patients. So what's now with the basal ganglia? This is how the regions of the basal ganglia look and their activity. We can similarly model this now inside the virtual brain simulator. This is the GUI, right? And this would be the first step, how to approach it, right? Look at the GUI, see what's going on there, see if you can replicate that, and do your own kind of little video of how this looks if we turn on the DBS electrode in the GUI. I will leave that out today. Sorry for that. I've guided in a previous year the participants through that thing. It takes about 70 minutes to get your own GUI simulation of a stimulus propagating through the brain. So I highly encourage you to go to our YouTube channel and find that personalized multi-scale brain simulation day 5 and check out that you can do it yourself afterwards. It's a really fun, evening activity because it looks really beautiful and you get a result rather fast. But now we will actually dive right in and look hands-on on the code. Yeah, so no GUI, because it's rather self-explanatory and in a previous video, but the Python interface of TVB. So this is how it should look in the end. We want to simulate a propagation of a certain stimulus ourselves. We will define a stimulus, we will visualize it, we will apply it to a region-based simulation, and we will visualize the results of that stimulus. Sorry for that. Okay, so what do we have to do? We log into eBrains right in the maintenance window. I know. Let's try it out and see if it works. Otherwise, I have a local setup and you have to do it later on or locally with me. We go into the Colab, we log in, and I've left everything like this in the slides, right? So you can do it later as well. And I will just do it right now together with you. Yes, all right, let's switch. Let's switch gears and go directly to a little hands-on. So I go to the TVB Edu drive and I find a notebook there. Let's see. Okay, it looks okay. Let's try it on eBrain. We go to the fall school, we go to week 8, and we see a notebook. We either download it just to be safe, or we can directly also copy to other libraries, my library. And I will do that again, because I know, so this is something you should follow along. The little arrow, copy, other libraries, my library. So what does it do if we submit? It just copies a version of this notebook to your library. So if we now want to go to the lab and test our notebook, just go on the left to the lab and you open in lab because it's a little bit bigger. Yeah. You select your favorite supercomputer. Okay, and here it seems, no maintenance. I'm in. I hope you're too. And here we go on the left side and find our library. So we go to that home folder to drive my libraries, my library. Again, home folder, drive, my libraries, my library. If you have copied it there, you will find it again here. Okay, then I have a lot of, don't worry. I don't know if you have so many here as well. If yes, well done. You followed along a lot of hands-on sessions. Not, don't worry. So here for this notebook, the credits go also to Anais, who has improved this notebook as well. So here we will do a region-based stimulation with TVB. So if we install everything, just execute this. If you are not sure if you have TVB installed, yes or no, and so on, it will just check and it will say, oh, everything's actually up to date. And I just, yeah, you can still see my screen. Okay. Otherwise, you let me know. Okay. Great. So we installed everything and now we load connectivity. We just load the default TVB connectivity. And it worked out, and we will check how many regions do we have, what kind of postulation do we have. Here we have 76 regions, and oh, we can check out the labels of that region. They're kind of cryptic, but you can look up in different papers, for example, the Sans-Leon paper or Spiegel et al. Papers. What do these abbreviations mean, like amygdala, amygdala, etc. So we will, in our case, select the left hemisphere primary motor cortex now, which will be region 50. And we will choose that one to stimulate. And for a stimulation, we will give a whole vector always in TVB that is defining for every region. It's as long as the number of regions, in our case, 76. We'll define for each region how much input will be given, this little I, yeah, the input to the region. And all right, so that will be in our case 0.1. We give a modest stimulus, right? There's no direct relation to the real world here. So if you want to do something, you might have to play with this number a little bit. And in our case, we are super precise. We can, in this, it's not the real world, it's the modeling world, right? So we can be super precise and only stimulate M1, the primary motor cortex. If you consider forms of stimulation, often it's more like the sphere that is influenced by the stimulus. So you are not 100% sure that you hit the right only then one region, but in our model we can do so, right? So let's define the weighting with everything is just 0 except for the primary motor cortex in the left hemisphere. And then we define the stimulus parameters. Because the stimulus is something bam, right? But in TVB, we can also define it a bit more elegant in a pulse train. Train because it comes back and comes back and comes back all the time. So the onset will be only after 1,500 milliseconds here because we want to have a long enough period in the beginning where everything settles and then so get into kind of resting state and then gives the stimulus. And we want a repetition of our stimulus every 1,000 milliseconds, and we want it to be 50 milliseconds long. So let's do that. And we also need to kind of configure the stimulus then. After we have defined these equations, let's give that as a temporal element of our stimulus. Let's give the right connectivity and let's give the right weighting. So we configure it, and in the same cell, we also want to plot it. So this is how it looks. We have here region 50, and it gets 0.1 stimulus. That's exactly what we wanted. And then it looks like a pulse train. It's giving different pulses, starts after 1,500, 50 milliseconds long, and it comes back again. And here you can see kind of a time space thing. It's that region 50 gets 3 inputs. And we loaded and defined the parameters of our stimulus and connectivity. Great, so we can already simulate. When we simulate, we execute here kind of a default simulation. And if you're not familiar with it, you always have to give these things and you have to configure and then you do sim run. That's a standard TVB procedure. So what things do you have to give? We talked about that we need a structural connectivity, that's here. We talked about that we need global coupling. In our case, that will be a difference coupling here. We talked about that these are differential equations. And how do you deal with differential equations? For example, from school, you integrate them to get the value, because it only describes a change over time, but what you want displayed is not the change, but the actual value. So you give an initial value, which is often just random initial conditions in TVB, and then your differential equation will do the rest for you, and you need to integrate it at every step here with a simple Hahn stochastic integrator function that will go over it and look always 0.5 milliseconds, will look what's the actual value of your state variables that you're interested in. We talked about monitors, and here is just a raw monitor of temporal average. So it's kind of like local field potential. We define the stimulus, right? And then don't forget, that's the important part up here as well, what kind of model we choose. We have seen the reduced one-Wang model by Deco et al. In our pictures earlier, but there are many more models that you can choose from, these so-called neural mass models in TVB. And one of these models, and I'd say nearly the simplest one, next to the linear model, of course, is a generic 2D oscillator. Simple, but beautiful and powerful. We will see that later on. And that model is has default parameters, except for those that you change. So here we changed 2 of the default parameters. Don't worry about it. You can play with it, put different numbers, try it out. And I've already pressed sim run, so we should have the results here. And this here is just plotting the results. And we can see how it looks, our kind of local field potential of all regions. So you can see a lot of black here. That's because every region has its own line here. That's kind of grayish. So when they're all overlaying, they look black. So the red one, the red line here, is giving us the average over all regions. And we can see we have a transient phase in the beginning that we want to wait. And then we see we have some responses to the stimulus here. Some are very high, low. So we see something's going on every time we get the stimulus every 1000 milliseconds here. So that's over all regions, and beautiful if you can see already a lot here. I don't. So let's visualize the stimulus on the brain. And the credits go to John Griffiths for this code to simply visualize our regions and their activity on the surface brain. So we need for that to plot it on the brain surface. Oh, I showed it. Let's keep it as surprise for now. What do we need to do? We need to kind of define a region mapping. Yeah, that's already inside TVB, so we just call upon it here. What does it do? It takes every region and it maps it onto the surface so that it looks beautiful on the brain, right? So if you don't do it, you cannot create beautiful brain surfaces with regions. So yeah, you better write some code like this yourself that you can visualize. And now let's see what it says. We will plot different times here, just before the stimulus and after. Okay, so just before, all right. And then we see already darker colors, meaning stronger reaction here, stronger firing rate, let's say, as the state variable. And it keeps on getting stronger, keeps on getting really dark blue here. And it keeps on going into the other hemisphere. We know we stimulated left primary motor cortex, right? But we see it spreads over to the other hemisphere, also quite a strong reaction there. So over time, there's not a video yet. One could also make a beautiful video. We will see some in a second. We have just programmed our first stimulus with the virtual brain, and we can now stimulate this simulate patient's brain, and we can see how it changes his or her dynamics, and if those dynamics somehow get closer to the healthy brain. And we can try out everything here with supercomputing backend, right? And with this kind of code. And we succeeded in doing so. I hope so. I did. I hope you guys there also succeeded in following along and getting something on eBrains or locally that looks similar, and that you get in the end those beautiful pictures of the stimulus propagation. We can do lots of things with it. We can choose different regions, and I encourage you to do so. We can look at different time points. We can look at the right hemisphere instead of the left here. There are so many things you can try out, okay? So what do I want to do? Maybe I want to, do I see I'm just checking if I see the chat. Yes. Okay, so I would like to choose a different region. I'd like to stimulate the amygdala, give like a fear stimulus. So which one is the amygdala? Oh, that's maybe too hard. Now I have to find out what's the number of the amygdala. Okay, I'm taking A1, region 0. And I'm saying I want a stimulus in region 0. Now let's make it stronger, 0.5. And execute everything again. Let's see. I don't know what comes out. Now it's simulating the same thing again. It's just stimulating another region, and this other region is stimulated stronger. It looks a little bit different. That's good. But it looks like we still have involvement of motor regions on the right. Okay. Okay. Okay, and we can go on with this for hours, but we will not. We will go back to our presentation and see what else we can learn and how we can make this kind of a toy simulation now more and more realistic. So let's see, we have 10 more minutes before the break. And we will look how we can make it more realistic a little bit and then continue after the break. So you have seen a mean field simulation of a stimulus and maybe performed it even yourself. And now I told you already that we have set out our project that we still continue by trying to bridge over single neuron spikes with mean field whole brain simulations. And so we wanted to build a multi-scale model to bridge the different scales. We have the spiking network on the one side, which will be a kind of the zoomed in scale, where we can look at the spike trains of single neurons, single excitatory, single inhibitory neurons. And that we will use for all the regions surrounding the DBS electrode to get kind of a close-up view of what's happening there. And then we also at the same time want to monitor the whole brain fMRI activity. And that's all possible with the virtual brain. So where did we start? We started here. We had collaborators with a previous publication on Parkinson's disease dynamics. And in this case, they used a spiking model of spiking neurons, all spiking neurons, no mean field, no neural masses, no the virtual brain. And they displayed the cortex with 600 excitatory neurons and an inhibitory loop with 150 neurons. And these are our collaborators with Fred Hamke et al. And Oliver Meier in Chemnitz. And they also displayed all the basal ganglia regions, the pallidum, the subthalamic nucleus, the striatum, and the thalamus with spiking neurons. Some were inhibitory, some were excitatory. And then they modeled this all, not with NEST, what you've heard from Dennis a lot about, I think 2 weeks ago in the multi-scale lecture, but with Anarchy, which is a short word for artificial neural networks architect. And when they modeled it with their neural simulator, they got out of these spiking networks some bold signal. And the bold signal was matching pretty well with their empirical data of Parkinson's disease patients, both signal and controls. So you can see here left is the experimental data, right is the simulation data. Here the same, here the same. These are just example patients and controls. And you can see that they had fits around 0.89 correlation between those. And those fits were making their model pretty accurate. And we said, hey, that's a cool model. We want it. We want to make it more realistic. And it's based on the Ezekiewicz model. And we just thought that we could replace just the cortical region, what they displayed as one node, with all the nodes inside TVB. Then we would have an input that is not just based on Poisson spike trains and noise and this one cortex, but it's based on something spatially and temporally more realistic. It's based on all the other cortical regions that are actually inputting into the basic ganglia. So that's exactly what we did. We replaced this one with the virtual brain, and we left the spiking dynamics to be the spiking dynamics, and we connected them, and we kind of translated them with TVB multiscale into mean field proxy nodes, and then we let them communicate in the whole brain network with the rest of the brain. And all the other brain regions that are not those, that are not the basic ganglia or thalamus regions, those were modeled with mean field models from the virtual brain. So this is our multiscale model. And these connections can be excitatory or inhibitory inside the spiking network. That's great about spiking networks. You can define many cool, very detailed things. But then you have also connections with other regions, and inside the virtual brain, everything is excitatory, all the connections there. So we have it all in one model. It looks quite complex. Maybe it will help if I show you a little bit what comes out of these models. So what comes out of the cortical regions is an average firing rate. So one line of the average firing rate for every region. We have seen that just in our simulation just before. And then out of what comes out of the spiking models for the basic ganglia and thalamus region is something that looks a little bit different. If you're not familiar with it, it might look scary. You need high resolution for this. These are so-called raster plots. And they actually have one dot for one spike of a neuron. So whenever you see here a couple of dots, we have here on the y-axis, you have the 200 neurons of that region. And when there is a dot, it means that this neuron spikes at this moment. So what it means that when we have a whole bar here, something like a black bar, it means that all neurons spike at the same time. And here you can see that the red line was actually the stimulus moment. So you can see, for example, the thalamus has enormous reaction to the stimulus. It just is completely inhibited, but then it starts with coordinated synchronized spiking. That was quite nice to find for us here when we stimulate the STN. All right, so we published this work last year in experimental neurology, and there are, of course, all the details for you. But I want to show, I think that's similar, just maybe a nicer output that you can see. We have the spike trains here, but also we can display it as raster plots. And here, this is in a very simple format, how we translate between the two scales. So in the one direction, we just count the spikes and make it an average rate. And in the other direction, we have an average firing rate in the virtual brain, and we just generate spikes. So this is kind of TVB multiscale in a nutshell, okay? And this is then scaled with a factor. It can be like a number, like 3, 4, called the interface factor. And between the TVB regions, we've talked about it, we have a scaling parameter G. Yeah? So it's just the interface factor is like a G, but between the scales. You can imagine it. So let's go into a little bit more detail here. And if you followed along the TVB multiscale lecture, great. If not, this is what you need to know for today. Is the virtual brain gives you mean field nodes. And these mean field nodes have one property per time point, which is the spike rate. Yeah? And this is then translated into so-called TVB proxy nodes. Yeah? Proxy, like in-between nodes, yeah, that replace those TVB nodes in our spiking energy world. And they will have exactly the same spike rate. And we, for arriving in our spiking network, we don't need rates. We need actual spikes coming in. Yeah? That's how the spiking network deals with it. It needs spikes, incoming spikes. So all we need to do here is generate spikes, which has the same spike rate. Yeah, and we did so, and this is how it can translate from TVB into anarchy. Just gives the spike rate, generates spikes that have the same rate, and it goes into the spiking network. The other way around, it's much simpler. We have spikes, but TVB wants a rate. So what do? We just count the spikes for a certain time interval, and this will be our spike rate. Now, the number of spikes divided by the number of neurons. We always scale with the number of neurons. That's a detail. Forget it. Don't forget it, but you know how I mean it, hopefully. Okay, so these regions are then connected with TVB via this spike recording monitor. A monitor that records spikes and makes them into a spike rate. Okay, that was the TVB multiscale part, which is well, the dense part. So let's give me 2 more minutes, yeah, and then we have a bio break. Because I just want to talk shortly about the underlying connectivity that we have for the model. Because we have the 2 scales, we also need kind of 2 different kinds of connectivity. One is a large scale and one is a small scale connectivity. So for this small scale connectivity, we used the connections inhibitory and excitatory from the previous network, and we embedded them, we put them in to our larger connectivity matrix, which was just a normative connectome based on the Peterson et al. High resolution atlas of the connections between the basal ganglia and cortical regions. So inside the basal ganglia, we have those inhibitory, excitatory connections, which are kind of the optimized weights that fit perfectly with one Parkinson's disease patient and one control here. And for the rest of the brain, we take a normative connectome. This is kind of a hybrid way to construct your connectivity. And just before the break, you can see a video of this multi-scale brain simulation. This would be the resting state period beforehand. Everything is fluctuating. Now the stimulus comes. And you can see that it changes the activity, of course, of the directly surrounding regions. But it also has an influence on the activity of other regions. Okay. So we simulate this, and of course in this video you can't see all of the results yet. So I will show you the results after the break, which will be in exactly 5 minutes. We will start again, and then we look at the results of such a multi-scale simulation of deep brain stimulation. For now, I thank you very much for your attention and see you back in 5 minutes. We have seen this video of how our multi-scale simulation looks in a video, but that is not what you publish in papers, and that's not what brings us insight. It looks beautiful though, right? This is what is then in the publication, which is firing rates. These are average firing rates of different regions of the striatum, the subthalamic nucleus, pallidum, thalamus, and for different conditions. What were the conditions? We had one control here, one healthy control simulated. We had one Parkinson's disease patient simulated, both in resting state. And then we also implemented a stimulus. And the stimulus was given to STN or to GPI. We tried out both here. To GPI, we just had a continuous inhibitory stimulus because the GPI is sometimes a target as well for Parkinson's disease patients to relieve them of their motor symptoms. And then we have the STN as a standard target for Parkinson's disease patients here at the Charité. And we tried out different stimuli. To the STN, we gave a DBS stimulus in a biphasic manner. You're wondering what's biphasic? That's exactly this. We give a pulse, and then we give a kind of opposite pulse as well. That is to not harm the surrounding tissue. That is what is actually given in the clinic, a biphasic stimulus. And then what's often in modeling given is just a monophasic, strongly inhibitory stimulus. And that what you look when you look at DBS modeling studies, this is what you often see. So we said, okay, we don't know, let's try it out, all these different kinds, and see if they have different results. And you see sometimes for some regions, this is control, patient, these are the three different stimuli. They are close to each other, and their influence on the firing rate is close, but sometimes different stimuli are doing different things. And this is all from the spiking model itself. And what is the output of the basal ganglia is the thalamus. That its activity is translated back to TVB, so it's important for us to check what's going on there. And we found a disinhibition of the thalamus through DBS. What does it mean? For the control, we see that the resting state activity was much higher than for the patient. And then for the patient, once we switched on the electrode in silico, we didn't stimulate thalamus, right? We stimulated another region, and somehow the effect was translated to the thalamus. And this somehow we will look at, of course. We see that the activity gets kind of normalized again, closer to a control activity, and making the patient, bringing him or her closer to the healthy regime. So that was in line with literature. There's lots of literature on spiking networks in Parkinson's disease patients, and they see this disinhibition of the thalamus. So it was great that we could see it as well. We could see it for the different stimulus forms. And this is how we looked at the average firing rate, and this is how the raster plots look for all the different conditions. So in resting state, you see a transient phase, and then you see kind of a rhythmic activity in all regions. And then you can see here the GPI stimulus is inhibiting GPI, and you see that it makes it kind of more. Here's a transient. Ignore that. So here is the GPI, and now we strongly inhibit it, and you can see that it's definitely spiking less here. So it has some regularity here, and then it's inhibiting thalamus as well. But somehow we have a disinhibition of thalamus, right? So it's leaving enough kind of gaps we've seen in our model to allow the thalamus to spike more. And we see for the biphasic stimulus, the thalamus is at first completely inhibited, but then we have synchronized spiking activity, and that drives up the spiking activity of the thalamus, making it closer, bringing it closer to the control. And for the monophasic stimulus, we see actually something similar going on, synchronized spiking in the thalamus after inhibition. We call this post-inhibitory bursting in the Izikowicz model. It's a phenomenon of the model, and it seemed to explain here very well what's happening. We have inhibition, but then this inhibition allows some gaps, and we have kind of this post-inhibition bursting. So more activity after inhibition. All right, so that's nice that everything is in line with the spiking literature, but the cool thing about our model, what was that? We could see both scales. So now we can also see the whole brain scale. At the same time, we can look what's happening there. So what's actually happening? We see in the resting state, when we compare patient and control, we see something blue lighting up. And blue here means that the control has more activity in that region, which is here the post-center gyrus, than the patient. So that is already a red flag, right? The control has more activity there than the patient. So how do we bring the patient closer? We want to activate that region. We want to make it more active and give it a higher firing rate. And you can see that we succeeded with the different stimuli because this region is turning red or even black for the monophasic stimulus and at least rosé for the biphasic stimulus, meaning that once we switch on in silico our DBS, we see that this region has higher activity than before compared to the resting state. So the stimulus is deep inside the brain, but here the postcentral gyrus is all the way on top and is still quite heavily impacted by such a stimulation. And then we see also that the frontal regions are activated, and they're all here quite red or dark red even, by our DBS stimulus. And we were asking ourselves, of course, is that normal? Is that something that people have looked at before? And we looked at our top 5 regions that were most increased activity after the stimulus, which was middle frontal gyrus was in there and the insula. And the middle frontal gyrus was actually found to have the biggest shift from DBS off to DBS on in an fMRI signal in empirical data. So that was nice. It's in line with ours. Actually, it's all about motor symptoms, but we see side effects, cognitive, and so on. And these could be explained by a strong connection and also then a strong influence on the DBS electrode onto the frontal regions here. It has kind of a direct way. There are lots of fibers going to the prefrontal cortex. So yeah, it's something we have to be careful if we want to activate that or not. And the insula is also strongly linked with non-motor symptoms in Parkinson's disease from literature. So we thought, oh, that's actually nice. And then we have something blue here that you might have seen before already. It's a bit odd that we have a kind of an inhibition of here the supplementary motor area caused by the monophasic stimulus. And then we looked into literature, and so supplementary motor area is known to be impaired in PD. And actually, TMS, so that's a non-invasive form of stimulation, it's transcranial magnetic stimulation of this region, improved freezing of gait symptoms of Parkinson's disease. So TMS here is like a coil, non-invasively placed against the head. We will cut to that. And that is directly targeting the supplementary motor area and is relieving other symptoms. So we were like, oh, okay, so it seems to be all connected and it seems to be all related to symptoms, what we find in our simulations. So as a first proof of concept study, we were quite happy with these summarized results that we constructed a virtual DBS model with TVB multiscale, and we produced realistic firing behavior in resting state and after different stimuli. We have the model in Anarchy, and there's also one version available in Nest, and we can observe cortical effects. And we are very flexible now with the individual model to test different stimuli. And we replace the cortex by something which is so spatially and temporally more realistic. We know exactly where the drive comes from towards the spiking network. And we know also when it comes and how it comes, and that it comes from realistic dynamics of all the cortical regions. All the code is available online, and we will look at it. But before we look at it, I have to thank for this study a whole lot of collaborators. As I mentioned already, our collaborators in Chemnitz, the founders of Anarchy, and then of course people from the movement disorders section, which helped with the data analysis and where the original fMRI data came from. And then my colleagues here, Dennis, Andre, Leon, Chin, and of course Petra for facilitating the whole project and being the leading PI here. The Ritum Consortium as well for the funding, of course. And now we took this model and we said, okay, it's a nice model, it's a proof of concept, we want people to use it, so how do we make that possible? And one way is making it first findable. And there's something called the eBrain's knowledge graph, and there we have an entry as well, where you find everything you need to reproduce the results of this paper. And I know it's everything because we had a previous master student contacting us from Italy, and she managed to reproduce all the results of our study based on what we put online. So that's the first success, right? Reproducibility, yay. So if you want to somehow go into that direction, or if you're just interested how one can make such a modeling reproducible, go on the eBrain's knowledge graph, I searched for myself. That's great that I copied that here. You can search also for the virtual brain or for basal ganglia or something, and you will find it on the model. And we have all the code on GitHub. And let me place the eBrain's knowledge graph link also in the chat, because it's often so much easier than going through the hassle yourself and kind of trying to, in a PDF, copying this. So here's the link to the eBrain's knowledge graph entry, where you find everything about it. And you'll find many more data sets there. Yeah? So whenever you're looking for a data set, check out this eBrain's knowledge graph. Maybe there's already data that you're looking for listed there. And then there are the authors, there's the publications. Sometimes it's already online, for example, animal data, or sometimes you have to contact the authors for gaining access. So let us now look at the code that was advertised there. And before we look completely at the code, I want to share the second link on GitHub. I will go there myself as well, which is a link that links you to a PDF. We've made this PDF called Reproducing the Results, where we also just in plain format explain there's the data, you have to set up this Docker image, you have to run it like this, so it's all containerized, and that you make sure that you have the same software versions, etc. That's why we put on the Docker online as well. And it explains also shortly, just in plain text, the different notebooks we used to get our results. So that's a nice resource to have. You don't need that now because I will guide you through everything. So that's why I already put now this third link, which is the main notebook for the main results. And I thought we will take maybe 7 minutes to look a little bit at the code. Yeah, it's TPB multiscale code, so it looks more complex than the TPB mean field code we have looked on in the hands-on session. But I think it's worth it because I will just point out a few parts now that might be interesting to you. So I put it up locally because then I also have the beautiful output pictures already. So it's directly in TPB Anarchy, it's then called. Yeah, it's a version of TPB multiscale that has Anarchy embedded. And you can see up here that you can choose different modes. You can simulate either the patient or the control. And also you can have different interface vectors here, but they're already optimized for you. But if you want to play with these, what I said is kind of the translation factor between the scales, this would be your factor. And then we have a simulation length, 1,500 milliseconds. We have a transient, right? That's all something that you know from TVB. And these are the initial conditions. Yeah. So here you can choose just in the first cell of the notebook already your simulation mode. So do you want to simulate resting state? Do you want to simulate the stimulus condition? And if you want to stimulate the stimulus condition, here you can give it in, do you want to stimulate GPI or STM? And then the simple stimulus is what we call the stimulus for GPI. It's just a simple inhibitory stimulus. And then we have bi for the biphasic, mono for the monophasic. Yeah, and all the stimuli, we gave them predefined already different frequencies, but you could also play with those. Yeah, it's 130 hertz frequency, and here 120, and between 120 and 130 hertz is also the frequency they use for actual DBS pulses in the clinic, so super fast. We have the amplitude of the stimulus, and we have the duration of the stimulus. You have seen the plots of the pulse trains previously. And then we say a couple of things that are configurations, we import a couple of things, and that's all in the first cell. Yeah, so you just have to change a couple of things to get all the results of the whole study. So the first step is always the structural connectivity data. That's why I started the lecture with that. I think it's super important to be aware on what everything is based here. And here we import the data, and this data is normative connectomic data, freely available online, non-identifiable, etc. And then we combine it with the optimized weights of the patient and control from the previous study of Meier et al. And replace what I showed with this hybrid framework. We replace inside the normative connectivity this little part of the basal ganglia for the patient and control with the optimized weights of inhibitory excitatory coupling inside the spiking network that were in the previous study optimized for fMRI data. And yeah, then we have also, you see here props, that's actually short for probabilities, because in a spiking network, just so you know, we don't only have weights and kind of the strength of the connection, but we can also define connection probabilities. So connection probability means, first of all, you can define there whether all nodes connect to all other nodes or whether you want to specify that. And probability here gives you an extra kind of handle on the connection. When you say, oh, only kind of in 50% of the cases, I want the nodes to connect, the others should not all be all-to-all connected. Because these are single neurons, and not all single neurons in one region connect with all single neurons in the other region, right? That's utopic. So we need to kind of put a more realistic picture. And that's what spiking networks are for. You kind of can get into little tiny details and get all to all those aspects that you are kind of summarizing when you use a mean field model otherwise. So we put all these together and we load also our normative connectivity data. That's all for just putting this hybrid connectivity together. And this is then what we get, a large connectivity matrix with all the regions we are interested in. And on the upper left side here, we have the basal ganglia regions well known now, like GPE, GPI, STN. And you can see that we have, for example, a strong connection here between the striatum and towards the GPE. All right. And we have the track lengths as well. And the track lengths, of course, are inside the basal ganglia and not so super long. It's all pretty much together there. But they can get really long in the other regions. Cerebellum, far away, kind of, from the motor cortex, for example. All right, so here we have this network only for the basal ganglia. Okay. I need to do a little bit of minus here so you can see it nicer. Yeah, so here we have the basal ganglia network that is kind of placed there. One for patient, one for control, comes from the previously optimized weights of the Meier et al. 2020 study. All right, so then you see already simulator. Now I make it a little bit bigger again. Here is where we import the co-simulator. We call it co-simulations, right? Because we, in principle, have two simulators running at the same time and exchanging information and a key activity. So once you call your simulator here, we put the connectivity just like we put it for TVB. We put the integrator just like we put it for the normal TVB, right? And we give it a step size, the integrator, we give it noise. That's all looking, and we have like raw data monitored. All right. So you see some aspects in TVB multiscale are looking very similar to TVB. We set initial conditions, we set delays. That's all pretty standard. And then we build and connect the anarchy network model. And this is, of course, a part that doesn't exist in TVB mean field simulation, and that now is important for our spiking network. So here it selects the nodes that are spiking. And in our case, we only have those 5 regions, basal ganglia and thalamus, as spiking networks. And then it calls the builder, which is the anarchy model builder, which is predefined, but you see the code below here as well. So you can just leave out the rest of the code here because everything is already in there. And that's taking into account the spiking nodes, our configuration so far, and giving you kind of the spiking network. But what is inside the spiking network, just so you see, it's like not magic or anything. It's the parameters that are here in common for the spiking network. These are parameters. You see V and 0 and 1. These are parameters of the Isikovich spiking model. And then we just copy them. We make always a deep copy and we update them for some parts. And this comes from the previous publication based on previous literature on these basal ganglia regions and animal experiments, where they have seen how do these regions spike, how do they behave. So we give all, like the striatum and things, we give them all single parameters. So it's totally heterogeneous in these spiking network models. Whereas in the TVB multiscale, if you've heard that before, normally it's all homogeneous. So we have the same neural mass model with the same parameters on every single region. What makes it heterogeneous and gives those beautiful phenomena is really the structural network, which is not everywhere the same, but which has different weights, different track lengths, and that's what makes it interesting. Though in the spiking network, we are the anarchy, artificial neural network architect here. So we are the architects of this network, and we can do whatever. In TVB, we can also do whatever, it's just not commonly done. So here we are going full heterogeneous, different parameters for different models. Oh, that was a little glitch on my computer. Okay. All right, and then just to show you one example of how we build a population is that we have certain parameters here predefined. We have our nodes predefined, and we put our population model on top of that. That's how you build this simulation. But now what I find interesting is how you build a connection in the spiking network. And you see, for example, that you do it like this, population connections. And for example, we have that a self-connection for GPE. So meaning that GPE is inhibiting itself with a self-inhibitory loop. And this is programmed with source is the GPE here, target is the GPE as well. The synapse model is predefined. We have a certain probability for this connection, self-connection, also possible in the spiking network. We give that connection also a weight. We give it a delay. And we also here can define the receptor types as part of the Isikovich extended version here. They have a specific GABA parameter. And you have different nodes as well that were also predefined. So these are the self connections. And we also have connections between the regions. We have seen the picture of it. And here it's just the picture written down with the source target nodes, or with source target. It's totally directed, right? This node inhibits this node. So you see it's a little bit more flexibility. It's a little bit more complex as well to define a spiking network, but it's also a whole lot of fun, right? You can be an architect here and you can make it super realistic. Once we have it, let's skip forward. We check again, we had 200 neurons for each of these regions. We also define our stimuli. We have seen the equations before. These are also from previous publications where we have actually the sinus form stimuli here. And the monophasic and the biphasic one are programmed here. Now we build the interface, and that's a translation, and you see it's all based here on rate. You can have different forms of translation, but in our case, we count the spikes and transfer rate. That's all we're interested in the end. Okay, and then we define from anarchy to TVB, from TVB to anarchy. We define a global coupling. That's now pretty standard. And reduced Wang-Wang excitatory comes in, because that's our neural mass model for the virtual brain here in that proof of concept model. It's for all the other regions. And you see here is a factor, and these are these interface factors that you need to define, which are kind of scaling up or down the connections in between the two networks. All right, so once you have all that, you see it's a lot, but don't worry. Often the spiking code is also available already in some publications, right? So if you are interested in a certain spiking network, maybe you don't have to invent it yourself and you can build on previous publications, just like we did. And we have so-called transformers. I think Dennis told you a lot about it on the TVB multiscale lecture. This elephant, funny, right? It's the software. Is really what makes this translation in between the two scales. The simulators need to talk to each other, which they are doing, and then we simulate. And we need to configure the Anarchy network for that, and then we just simulate. And I just want to skip forward to some results where we have some results on the TVB side, which you can see here. It's again a state variable of the reduced form Wang model. And you can see that you have per region one average firing rate, for example, or here it's synaptic gain, but don't worry. And then at the same time, out of your simulation, you also get, for the spiking nodes, you get their activity averaged. But now here comes the beautiful part. You also get full-on raster plot. Yeah, and you can see in this simulation what every single neuron did. Either synchronized or unsynchronized. You can see exactly what's happening. This is a resting state simulation, so we don't have a stimulus here. That's why it looks so regular. Once you have a stimulus, everything is disrupted. We have seen that. So if you set up the Docker and play with this, you can always contact us if it didn't work out the first time for you. Then you can see many, many different phenomena. And for example, synchronization is a phenomena that's also well known to Parkinson's disease that is over-synchronized sometimes, and you kind of want to disrupt it. So you can take this model and you can play with it and see how you can get less synchronization inside the basal ganglia regions and kind of, heal your virtual patient. Yeah, so it's all available for you. And you can set it up, run it locally. You can also run it on eBrains. Dennis has explained how you can run TVB multiscale on eBrains, also possible. So we have looked at the code into a kind of super realistic multiscale model where we see the different dynamics of the basal ganglia regions very, very much in detail, and of the other regions kind of from further away in their average activity mode. And now, where are we? How can we improve the simulation in the future? So I want to take a couple of minutes to share with you unpublished results that we are working on right now, finishing up and publishing, where we try to make this model even more realistic. So you're wondering, like, how is this possible? Yeah, we forgot one thing in the last model. It already was super detailed, but the placement I told you about earlier was not yet considered. We still activated the whole subthalamic nucleus or the whole GPI once we gave the stimulus. All the 200 neurons received uniformly distributed stimulus. So that's not realistic. We know in reality, we have an electrode placed in 3D space. And using the software LeadTBS, you can actually reconstruct with a pre-operative MRI and a post-operative CT. You can reconstruct these pictures and see exactly where your electrode is placed and bring it also to MNI space. And then we want to combine that somehow with TVB. We want to somehow input that super detailed 3D information into TVB. And there was one previous study who has done so. That was by Sora An et al. From 2022. They looked at the fiber tracts, every single fiber tract, and they integrated that into a neural mass model simulation, into a brain network modeling. And they used a surface-based simulation. So if you have not heard of surface-based, you can take every region and put a neural mass model on top of it. But you can also go into more detail and take every triangle on your surface, put lots of triangles on your surface, and put on every little kind of voxel on top of the surface. You put a neural mass model, giving you more like 16,000 nodes in your network, but it's possible. And then you can connect those triangles with the fiber tracks, and you can then stimulate exactly one point in 3D and look at the electric field around this point and look which fibers are crossed or which fibers are crossing that electric field. And you can activate that and then even look at how long is the distance between the activation of my fiber tract and the final destination, and consider that for your distribution of voltage along your fiber tract. So it's like on top of a surface-based simulation, also considering the stimulus input in terms of every single fiber. And they have used data from treatment-resistant depression patients, 3, and they have found the yellow parts are quite high correlations when they checked their evoked responses on EEG with the empirical data. So let's have a look. Oh, they're talking for, we could just watch a video, right? These are simulated evoked response potentials by DBS, here placed in a totally different region, SCC for depression, different targets, when they're switched on and off. And you can see here is the stimulus start, and you can see on the EEG signal here that it has quite an impact on lots and lots and lots of brain regions. Let's watch again. No, that was the end. Comes again. You see, different contacts are activated. An electrode has different contacts. We'll come to that. You can choose which contact to activate, 4 different ones. And here is the first picture of a simulated response to the stimulus and versus the empirical one. And you can see here, this plot compared with this one, the time delays and everything does not seem to be the same yet. There are some differences, but for some, we have quite a high correlation of like 0.4 with the empirical data, which was for us like, oh, cool. So there is some possibility to connect somehow the exact 3D location with the fibers with the TVB simulation. So we said, let's do it. Let's do the same. So we take the reconstructed 3D location of the electrode, we calculate the electric field around it in 3D space, and then we have all the fibers from a normative connectome, and we calculate the intersection of that field here in this red with the fibers. And when a fiber is activated, we check which fibers are activated once they are above a certain threshold, yeah. And then we trace them up to the cortical regions, and we say this fiber is activated, and this region has like 10 activated fibers arriving. So I'm giving a 10 stimulus into a TVB simulation to mimic the stimulus activity arriving at that region. So we did it in the end here. Region-based, not surface-based, that's a big difference with Ann et al., but we adapted the approach from Ann et al. So when we look at this, and here is the activity of K1, so one contact in our electrode, and here we choose a different contact in the electrode. So contacts are millimeters apart, yeah? So you see here in the middle that there is some difference when we activate one or the other contact, meaning that see the middle, lights up. That's the difference. It means that in our simulation, these are now sensitive to the exact 3D location of the electrode. And to give a cherry on top here, I want to show you that now our simulations are super, super, super detailed and high resolution, right? But now they also seem to fit with empirical data. And this is in such an electrode, and they have performed different experiments activating different parts of contacts, so-called contacts of this electrode. And in the middle of these two contacts can be activated as a ring, the hole, or they can be activated kind of like a pie with three parts. So they can be activated in a directional fashion as well, called ABC. So the A contact, B contact, or C contact can be activated alone. So they took Parkinson's disease patients and experimentally checked different contact activation and always measured whether their motor skills were improving or not. And 100% is when they're the same. And if they're above 100%, those motor skills have improved by activating one of these electrodes. So here you can see actually data of more like 14 patients, and each were having different activations. And we simulated these evoked responses by the stimulus. And you can see a positive correlation here, significantly positive correlation of predicted and empirical motor improvement. And the prediction is based here on TVB simulations with this high resolution stimulus input. So that is quite promising to us. We're in the process of publishing that paper, which relates for the first time TVB here with clinical scores of Parkinson's disease patients. For that work, I have to make sure to thank a whole lot of people here involved, among them many of the colleagues that you might see in other lectures, and also the movement disorders team from the Charité who provided the data for that. Especially I want to thank here again Petra Ritter as the PI of this whole project who made this possible. And it's here just a QR code, which is to a previous YouTube tutorial where I tell also everything about how to reproduce our former study. So now I put in just a quick outlook. We have a couple of more minutes. So a quick outlook of where are we heading with this whole brain stimulation. And there are a couple of newer developments. So one is the closed loop system of brain stimulation. So when we find a certain feature, for example, in our simulation, that predicts improvement of motor income, we could measure that as well. I said there are now electrodes that are sensing, right? So we could measure such a feature that we find, and we could measure it in real time in a patient's brain, right? And through some app or something, right? We could see when that feature is there, we need the stimulus on. When that feature is not there, we don't need the stimulus on. The patient is doing fine. And that feature that was mostly explored now and is implemented in such a closed loop is actually beta bursts. And beta power is sensed in the electrode and is taken as a control variable to give feedback and set new parameters for the electrode. So there are many studies now upcoming that are looking into this new generation of electrodes, which can sense as well. And this sensing is giving us a great opportunity to close the whole loop and to actually make our stimulation not only personalized and find the right setting for an individual patient, but also time-dependent personalized. Yeah, so maybe I had a funny paper on that as well, that during Christmas things were totally different for these people. But okay, that's you can look that up, pretty cool. By the same authors here, Andrea Kühn, Julian Neumann, etc. All right, so this gives us new opportunities, right? And they have already just published, just 1 month ago, a Nature paper where they had such sensing electrodes implanted for a longer time in patients that were suffering from treatment-resistant depression. Yeah. So then they were looking, the team around Helen Nyberg, they were looking at what is actually happening for those patients over time. And the things that we can measure is the local field potential from the electrode, from the contacts of the electrode. And they build a machine learning model with a neural network classifier to classify and predict what will happen to the patient. And they call this, what they found, a spectral discriminative component, which is kind of a combined feature of different things that could be measured with the electrodes in different spectra, frequency spectra. So when they look at this feature over time for their patients, and they left one patient out of the algorithm, so was not trained on the algorithm, they actually found that it was possible to predict the depression relapse of that person approximately 5 weeks before clinically there was a relapse. So these sensing electrodes give us the possibility to predict the future, if you want so, for patients, and maybe then determine with some higher order features. Here's machine learning of the sensed signal on how this patient can be helped even earlier, how we can do like, a priori, before something is happening, how we intervene and maybe change stimulus settings. Yeah. So I find this pretty cool. It's a cool paper. Check it out. That what would the future bring in brain stimulation? So what other things are there? Yeah, just shortly. There's focused ultrasound. There's lesioning, it's traditional, right? And there's also this non-invasive form, transcranial magnetic stimulation. And you see the FDA approvals for these different stimulations, more and more upcoming for so many different diseases, like neuropsychiatric diseases, neurological diseases. So it seems to be like a whole wave of new treatments coming into neurology and psychiatry. So what is transcranial magnetic stimulation? Very shortly, it's non-invasive, so you don't need a surgery, right? And there's this new protocol also for TMS or repetitive TMS, that you receive repetitive TMS for, I think, 37 minutes for 3 weeks, always 5 days a week. And it showed that significantly patients from treatment-resistant depression are feeling better after those 3 weeks receiving this treatment with the magnetic coil and stimulating non-invasively. So we see it works, and to understand how it works, we need modeling, I think, and many people think so. And there's also another TVB study already out there that I should mention, is by Kurz et al. It is already a couple of years old, but it's the first study that virtually uses TDCS, which is also a non-invasive form. It's transcranial direct current stimulation, in a model, and checks and compares the effects with literature and finds quite a nice overlap. So that's also already out there. Coming back to our big picture, yeah. What have we seen? You've seen in lectures that pharmacological interventions can be simulated with the virtual brain. Now you've seen that brain stimulation can be simulated as well with the virtual brain. So this is just a fun slide of the future, how we can imagine it. So maybe you know the neuralizer. Anyways, so that's a device that completely lets your memories vanish of the last couple of minutes. Yeah, that is not so far in the future anymore. And also the Neuralink you've seen is evolving super fast, and they want to connect it with AI. They want to make big steps there. So you should all closely follow it. Maybe these kind of sci-fi ideas are not as far away as we think. Now, this is a prototype that we thought of for the future that we would like to have. We upload MRI onto a browser. We upload the reconstructed electrode location, and we generate and fit a virtual brain just by a click here. It's a mock-up, right? Not working. And we look at the activity. And then what we would love to do with the virtual brain setup of DBS is give in different amplitudes, frequencies, and test those stimulus parameters, and always show again the dynamics to the clinicians. And this could, for example, work on e-brains, with a supercomputing backend. And then we could also click to optimize the parameters and provide us a likelihood of improvement for each symptom. So here, for example, comes out that the algorithm found we should have a 2 milliampere amplitude and a frequency of 130 hertz. Credits here go to Dinara and Timo for setting up this mock-up, how we imagine it could look in the future for modeling assisted clinical interventions of deep brain stimulation. So what do I think of the future of DBS? And this is already the last slide with content. I think adaptive DBS will be a thing. So adaptive in the sense that we have a closed loop. We need to find features and extract features that we can compute in real time. And I think definitely we should move towards non-invasiveness in general, because DBS surgery carries a lot of risk, and we have seen also great results with non-invasive treatments. So from especially the patient side, the non-invasiveness should be explored first and foremost. I think it will be modeling assistant. Will there be real-time computation of AI features to predict patients' relapse? I think so. We will have individual level solutions for kind of different apps that work, different algorithms work for different patients. Hopefully a multimodal monitoring and also longitudinal monitoring of these patients, because once you find the optimal setting, it doesn't mean that this optimal stimulus setting will be the same in a couple of years. It needs to be adjusted continuously. And of course, I think it will go towards multi-scale modeling. And the cool thing I found when I was looking for new things, I heard that there was, anyways, there's now things called like nanoparticles and nanoelectrodes, and maybe you've heard of nanomedicine, super cool. And they're exploring that also for stimulation. So meaning that there's already some things out there, and they've tested it successfully in mice, that are super small devices. They don't need charging. They are kind of feeding off the electrical impulses that are already in the brain. How cool is that? They're to be developed for humans, and they will be then entering through blood-brain barrier. Crossing that, they will be somewhere in your brain. They will be super small, and they will kind of repair connections that need continuous stimulation. And then you can also activate them from the outside with TMS if you want stronger responses. So there are all kinds of these ideas out there. And they can also connect to certain parts in your neuron. So there are different nanoparticles for different parts and different nanoparticles for different receptors, for example. So that's super cool. And it's not sci-fi. It's like it's happening. Yeah. All right. So I think that will be the future. And then we will need models for the nano level, but we still want to use whole brain and see that we also capture that scale. So we need more resolutions as the brain has more resolutions. Okay, just a short slide in case eBrains didn't work for you. Don't worry, you can go back and find this manual that I wrote, or this short intro of how to run everything locally. And here as well, if you want to know more and want to know some references, here is more. We have given previous courses on similar things on brain stimulation. I have, Dennis has. They're all on our YouTube channel. And with that, I want to thank you very much for your attention, 2 minutes over time, and open up the floor for questions. And if there are no questions now, always feel free to contact Petra or me or anyone else of the team for answering hopefully all your questions on brain stimulation and how to simulate it. It was a pleasure for me, and I hope you enjoyed as well.